first, thank you for coming. My name is Janna Fierst, and I'm at the University of Alabama right now. So one of the major interests of my lab is the influence of sex on horizontal gene transfer. And we've been trying to study this in assembled genome sequences. But what we found is that it's actually really difficult because of the level of contamination that occurs in most assembled genomes. So first, it's a problem because you can look at contaminants and think that they're true horizontal gene transfer. But what we've found is that there's not really any good bioinformatic methods for actually eliminating these contaminants. And the ones that do exist actually risk getting rid of true horizontal gene transfer because they're basically looking for everything that looks like a bacteria or a virus, and that's what horizontal gene transfer is. And so what we've been working with then is machine learning techniques. And so what is machine learning? Generally, it's when we're asking computers to recognize patterns without explicitly telling them what those patterns are or what they should be. For supervised machine learning, we construct a model given a set of labeled data, and then we use that model to classify future observations. So here, for example, we're looking for DNA that we've labeled as either foreign or as target. And so what we've been working with is actually a method that's called a decision tree. And essentially what it does is that it asks a series of binary questions out of your data, and it attempts to either then class them into different categories or ask future questions to go forward with the classification. But the most important thing about a decision tree is that it actually is able to tell us which are the significant factors, which ones are diagnostic. So we measure 20 different things about our assembled scaffolds, we load it into the decision tree, and it uses an information criteria to tell us what's actually diagnostic. So for example, this is some of the work I did during my postdoc. We were looking at Caenorhabditis romanii, which is a small nematode worm. And so nematodes are filthy. They live in the lab, they eat bacteria, they're covered in bacteria, they have it in their guts, parasites of bacteria, everything comes out in the DNA extracts. And so what we did then is we used BLAST to identify a set of training data. So it's scaffolds that we knew we could associate to Caenorhabditis worms in some way, and ones that were blasting to bacteria, other organisms. When we ran the decision tree, what you can see is that the final assignments were able to pretty robustly separate out the foreign DNA from the target DNA based on two variables. So the first is the scaffold GC content, how much GC versus AT is there actually in the assembled sequences, and then the second is the per base sequencing coverage. So different contaminants are going to be at lower coverage, some are actually at higher. And so when we look at the decision tree, that's what you see. It's basically asking a series of questions, carving out a model around these two variables. And so the next thing that we looked at then was an organism with a lot of horizontal gene transfer, because we wanted to know how this would actually affect the model. Would it be that true horizontal gene transfer is so complex we can't find the contaminants? What would happen? And so what we looked at then was the Videlid rotifer A. vega. It's asexual, ameiotic, and it's estimated about 8% of its genes are actually horizontally derived. And so when we did the BLAST for these, what we found is that the contaminants here are actually mostly human, and then some E. coli. And then the A. vega are the ones that don't blast anything, because there's not a lot of rotifer sequences in the NCBI databases. And so when we ran the decision tree, what we found is that we could identify the foreign DNA, but it's much more complex now. You can see that the foreign ones are actually sort of nested in with the A. vega DNA. And when we look at the decision tree, it's much more complex as well. So now we've got 5 factors that are diagnostic. So we've got the average sequencing coverage, also the GC content. Now we've got scaffold length, a measure of how well things are assembled, the percent of read covered in realignment, so how well does your read data set actually match to your assembly, and then the RNA expression, because these contaminants were found in both the DNA and the RNA. Okay, so thank you for listening.